We're going to go through this schematic of specific immunity, and that is the immunity which revolves around the specific antigen sequences of polypeptides or glycoproteins or glycolipids or polysaccharides that are found on pathogens that invade our body and have got past our initial barrier defences. And the thing about the specific immune system, or it's sometimes called the adaptive immune system, is that it revolves around B and T cells and ultimately the production of antibodies which are produced by a special type of B cell, which results in phagocytosis and digestion of pathogens. And that happens through a number of immune cells talking to each other. And that is what this schematic is showing. And so I thought I'd quickly go through it. It starts off with these cells here. And these cells are the antigen presenting cells of dendritic cells. This is a dendritic cell with all of the arm-like appendages coming off. And then there's a macrophage here, which you can see, which is in the process of ingesting some yellow bacteria. And these guys do something really clever, because when a pathogen gets into our tissues, it will end up coming into our lymph system. And the lymph system is like a blind-ended tubular system, so it's not a circulatory system that runs behind our circulatory blood system. And the tissue fluid and the plasma of our blood seeps into this lymph and it circulates. And I've just described it a bit like a swimming pool filter system, where all of the soluble parts of the blood, including some pathogens which are tiny, get into this lymph system, and then they drain through lymph nodes which are found within this lymph system. There's tens of them around, and that is a little node which is like a concentrated set of lymph vessels where the immune cells are particularly concentrated, because they know, like a swimming pool filter, that the pathogens are going to seep their way towards them after they've entered the body. And it's like a filtering system. And so our dendritic cells and our macrophages are going to do this very clever thing of engulfing and digesting a pathogen. And then instead of entirely digesting it, it is going to partially digest it and then present on its surface a section of the pathogen called an antigen. And this is a recognition of a non-self antigen by the macrophage and the presentation to the rest of the immune system, which is a bit like saying, everyone, I found this, it's not us, it's non-self, and I've digested it and it's in my MHC hands, is anybody interested? And the macrophage, or the dendritic cell, will hold that antigen in the MHC2 and wait. Now, in the lymph nodes, you have these dendritic cells and macrophages, and you also have T cells. And T cells are, and specifically T helper cells, have their own receptor. And we can see one here. On the outside of them, they have T cell receptors, and they are of a complementary shape and variation, which matches a certain antigen sequence, a polypeptide sequence or polysaccharide, as I've said, glycoprotein, glycolipid. But importantly for T cell receptors, it matches a antigen that is presented in the MHC2. And so you would have a piece of antigen, and that will be held in the MHC2, and the T cell receptor would be complementary to those two together. Okay, and that means they can bind, and we can see here on this diagram that this is the binding process between the MHC2 holding the antigen and the T cell receptor, which is complementary in shape. One thing to say about this is that the binding takes place as a result of a number of binding proteins, and you can see that it forms a complex. And this is a bit like the way that a transcription complex forms with a number of different proteins. And so you have a number of proteins here which will form a binding. One thing about T cells, and that's not shown on here, and I'm going to draw it in on this particular diagram, is that they have what's called CD4. And CD4 is a protein on the T cell which helps the binding to MHC2. The reason I mention CD4 is it's going to be very important when we study HIV, because the CD4 is something that the HIV latches onto, which is why it's capable of attacking T helper cells in our immune system. So where have we got to here? Well, we've got a macrophage and a dendritic cell which have presented antigen in their MHC2 hands, in inverted commas, and the T cell receptor, which is complementary to those two, has bound. And we are then going to have T cell activation. And the T cell activation is going to happen as a result of the macrophage producing, you can see here, these cytokines. Cytokines are chemicals of the immune system, and it actually says IL-12, so interleukins is actually a word. And they produce these, and the interleukins are going to go and stimulate via receptors on the surface of the T cell to do two things. The T cell is going to actually activate the macrophage. So there's a bit of kind of like a symbiosis here to become more aggressive and go and phagocytose more of the pathogen. But it also, and this is quite clever, it releases cytokines which actually have an effect on itself. And that causes a mass proliferation of this type of T cell with this type of T cell receptor. Because pathogens don't go round in twos, they go round in thousands, tens of thousands, hundreds of thousands, even millions. And therefore, when we have found a certain antigen in our lymph system via the antigen presenting macrophages or dendritic cells, we don't just want one T cell to go out as some kind of lone maverick trying to find them all and kill them. It can't. So we need to enlist a whole army of that type, and that's why it's the specific immune response, that type that is complementary to that antigen. And so the end result of antigen presentation by the macrophage or the dendritic cell is we've got T helper cell activation, which has led to millions of that type of T cell now being produced. And so there they are now floating around in the lymph, and they're floating and they're looking and they're waiting until their T cell receptor might come across another complementary MHC2 and antigen. The immune system will not disappoint, because this is where B cells come into play. B cells are also in the lymph system, and what happens with B cells is you can see here on their surface they have membrane-bound antibodies, call them immunoglobulins, and there are many on the surface of the B cell, and they are floating around, and they will have come across the pathogen, and they themselves will become antigen presenting cells. So they themselves will actually do this process. But instead of being a macrophage, the B cell endocytoses the pathogen, it partially digests it, and presents it on its surface with MHC2. And you can see this is one that's where it's been done. So the MHC2 is here, and it's presenting the antigen. And just like the T cell binding to the macrophage, a T cell with the complementary T cell receptor is going to bind to the MHC2 and antigen complex. And the binding of those two together is going to cause the T cell to release chemicals which are going to activate our B cell. And this is really, really important, because this is going to give us a flooding of the fluid systems of our body, the circulatory system, the lymph, with antibodies down here. So we're going to get to this stage, and we're going to get to it because the activation of these B cells leads the B cells to turn into what we call effector cells. And effector cells are plasma cells. Plasma cells, they they really take our ability to fight pathogens to a new level. Because if we go back, if we look at this B cell, you may remember that I mentioned that the antibodies on it are all membrane bound. That means the ability for an antibody to interact with a pathogen, and the antigen on the pathogen, is only as successful as a whole B cell bumping into, colliding with that pathogen or that antigen. Well, the effector plasma cells, they release these guys, soluble antibodies, and these antibodies, tens of millions, perhaps even billions produce, flood the lymph and the blood, and they are free to move anywhere. Okay, if you've seen Lord of the Rings, I actually think it's a bit like the army of the dead that he unleashes, where they just run through the area and just kill everyone, and because they're indestructible and they just go everywhere. And the ability of antibodies to flood the lymph and the blood system means that the pathogens, there's nowhere to hide. There really is nowhere to hide. And so they, within those systems, the corresponding antibody which matches the original antigen means that you're going to get antibodies binding to pathogens throughout the whole of the fluid systems of the body. And because it's the fluid systems, we call this the humoral response as well. In the olden days, I think Greek or Roman times, they used to call your fluids your humours. And so the B and T cell response is the response that runs through the fluids of your body. And the free-standing antibodies, the billions and billions of them probably that are thrown out there, mean that there's no real safe place for a pathogen. Now, new slide here. What do the antibodies do when they are flooding these liquid parts of our body? And there are a number of ways that antibodies bring about the destruction of pathogens. And this slide here gives us these. The antigens themselves do not result in the destruction of the pathogen. And the ways that they are involved is by various processes which lead to a more efficient destruction. So the first here is if we come to neutralization. Neutralization is the antibodies binding to receptors and binding sites of bacteria or viruses, which means that they can't dock into the host cells. They can't show their pathogenic tendencies because they can't actually bind to any of our cells. So they become inert, they become useless, and they're just floating around. And therefore they're just floating around, and that gives us time, which means that if a phagocyte comes along, it's going to engulf and digest the pathogen, which is a bit of a sitting duck. And we call that neutralization. So number one is neutralization. Number two is agglutination. And agglutination sounds a bit like glue, and that is exactly what happens. The nature of an antibody, if I come down here, it is a Y-shaped molecule with two variable regions, number one and number two. And that means that the same antibody is capable of binding more than one antigen, which is on a pathogen. And so you could actually have the same antibody binding a pathogen here and a pathogen here. And that means that in a complex of antibodies and pathogens, you have them clumping together, agglutinating. And the great thing about that is that when they are all clumped together, it means that when a phagocyte comes along, it is capable, and it would be a big old phagocyte, it is capable of engulfing and digesting large numbers at the same time, all conveniently packaged together. It's a bit like if you've seen those adverts for consolidate all your individual loans into one easy monthly to pay loan. They're all there waiting to be phagocytosed because they're all stuck together. Brilliant, and a very improved and efficient way of doing phagocytosis by agglutination. Number three is going to be, you can see here it says precipitation, and precipitation, it it's a bit like agglutination. We don't really need to know about this one, but it's more that it just brings them out of solution and makes them easier to phagocytose. The next one is they activate the complement system, the antibodies, and that means that they are able to punch holes in the membrane of the foreign cell, and that will lead to lysis. Lysosome means to electrolysis is splitting with electricity, and lysis means to break or split, and it involves the death of the cell. The one on here that isn't here is the enhancing of phagocytosis by, and it's an important word, opsonization. And opsonization is where we have an antibody which is bound to a pathogen, and the constant region of the antibody is actually complementary to a receptor which might be found on a macrophage. And so that creates a shaped base complementarity between the antibody and a receptor on the macrophage, which is almost like a docking station. And so therefore it increases the efficiency and the chances of the antibody docking, and therefore this whole antibody pathogen complex will be endocytosed and digested, and that will lead to the destruction of the pathogen. And so we call that opsonization. So the ones that we really need to know about at A level are neutralization, the binding of on receptor and binding sites of the antibodies, agglutination, which is the sticking together of pathogens into one easy clump that can be phagocytosed, and opsonization, where we make the phagocytosis more efficient by the antibody having almost a docking station for the phagocytes to increase the efficiency of phagocytosis. And so those three in general are the ones that we would need to know about antibody action.